So hello, everybody. Welcome to this webinar. Thank you for joining. So I will, in my talk, talk about oligodendrocytes and myelin white matter, give you a short introduction into the topic before I talk about remyelination. This is the process of regenerating myelin after it has been damaged, and particular focus on the role of lipids in this process. So let me start by showing you a human brain. So the human brain contains gray and white matter, and what you can see is how extensive the white matter is of a human brain. So it occupies large areas of the brain. Now, the white matter is white because it is highly enriched in lipids, so it's a very fatty organ, actually, the brain. So after the adipocyte tissue, the second richest organ with lipid composition. So what are the main components of the white matter? So they are oligodendrocytes are the main cellular component, first identified by Ortega, by silver carbonate staining. And you see this cells with these fine processes. And each of these processes at the end ends up around an axon. And with the invention of electron microscopy, one could see this beautiful structure of this multi-layered membrane around the axon. Now, myelin has a quite particular structure. So it's, as I already mentioned, it's high in lipids. Around 70 to 80% of its mass is lipids, and there are few structural proteins like the myelin basic protein and the proteolipid protein. Now, it's not only high in lipid, it also has a specific lipid composition, so a ratio of cholesterol, phospholipid, and glycolipids to 20%, and it has some signature lipids like galactosylceramide and plasmalogens. Now, one protein, the myelin basic protein, is a key structural protein that is required for myelination. So it's a protein that is locally translated in the myelin sheaths. It's positively charged, and when it interacts with the negatively charged leaflet of the myelin bilayer, it starts to polymerize and to compact the myelin sheath. So this can be seen here. So you see in yellow the myelin during myelin growth, where myelin still contains cytosol, and you see how myelin compaction starts from the back and progresses to the front by the activity of the myelin basic protein. So it functions as a zipper, closing myelin, and at the end leading to this multi-layered stack of membrane. Now the result is stretches of myelin that are arranged along the axon in a characteristic pattern. So these stretches are interrupted by small gaps. They are called node of von Weir, where the action potential is generated. And by this arrangement, the action potential can jump from one node to the other. And this is the basis for saltatory nerve conduction. And this makes our nerves much faster than they would be without myelin. Now, a second important function of myelin is, so once the axons have been myelinated, they become dependent on oligodendrocytes and myelin for trophic support. So, for example, molecules like lactate and pyruvate are transferred from myelin to the axon to fuel the axon with energy. Now, I will not in this talk about the process of myelination during development, but I will talk about the reformation of myelin in diseases called remyelination. So one of the most important diseases in this respect is multiple sclerosis. So this is presumably an autoimmune disease where you have an autoimmune attack, an inflammatory attack against the myelin sheath. The antigens have not been identified, but the result are demyelinating lesions that form in different areas of the brain. So here you see at autopsy areas in the white matter that appear black now because they are demyelinated. Now, in a histological, neuropathological examination, we see here a staining of myelin in blue, and then you have this sharp border of the lesions and areas that are completely devoid of myelin. Just quickly to review the process of how these lesions evolve. So when the lesions form, they're called active lesions. And the main cellular component of these active lesions are myeloid cells or macrophages that have infiltrated into the brain or resident microglia within the lesion. And there are a few lymphocytes. Now, there are different possibilities of how these lesions then can evolve over time. So they can lead to chronic active lesions, shown here in the upper right corner, where you have resolved inflammation but no repair. And then there's a second possibility. This is that the lesions indeed do repair. This is then the remyelination, where you see these pale blue areas as a marker for reformed myelin. Now, this is not very frequent in humans, but at autopsy, you do see lesions that have regenerated. Now, the worst outcome of an active lesion would be chronic active lesions. So these are lesions where the inflammatory cells still remain in the lesion, and they are found at the border, at the edges of these lesions. Now, these lesions, this chronic active lesion, they correlate with progression, and they can even expand and enlarge. So one key question we have is to identify factors that determine lesion recovery. So at autopsy, some patients have more or less remyelinated lesions. We want to understand what are the factors that determine when lesions repair or not repair. Now, a model that we are using in mice is the stereotactic injection model, where we inject a toxin into the white matter, and this leads to a demyelinating lesion. So after 4 days after the injection, we have lesions, and then we can follow the repair which occurs within the 2 weeks after the lesions have been induced. So this is how it looks like. So we have here now a staining of myelin in green. So you see this demyelinating area in the white matter. Now after a very short time, this lesion is populated by phagocytes. So these are cells that have migrated into the brain, but there are only very few of those. So this monocyte-derived macrophages. So the main cellular population are the microglia that start to proliferate within the brain. So the microglia are the macrophages of the brain. So how does this occur? So when you have a demyelinating injury, the question is, how does the tissue know, the cells know how to regenerate? So one key element here is the innate immune system. So microglia are the main cellular component of the innate immune system in the brain. And these, so there must be sensors on these cells that sense the damage, and then the sensors translate this information to an effector program, to transcriptional response that then leads to repair and instructs this repair program also to other cells like oligodendrocytes, precursor cells that then start to differentiate and to myelinate. So what we really want to understand is, how do cells know how to repair a lesion, and how do the cells communicate in this process? So just to review briefly the function of the microglia, the phagocytes in a lesion. So as I mentioned, a few days after the lesion have been formed, they are full with phagocytes. Now, the myelin debris that arises from the damaged myelin is found in the extracellular space, and there must be receptors that recognize this myelin debris. So these are scavenger receptors, and we have found that phosphoserine receptors, all of them can identify and interact with myelin. Now, the myelin debris has to be phagocytosed into the cell, and now in the lysosomes, the membrane and lipids are broken down and degraded. Now, there's the key component of myelin, or the main component of myelin, in fact, is cholesterol. And cholesterol cannot be degraded. There are no enzymes to degrade cholesterol. So cholesterol has to be stored in lipid droplets as cholesterol esters. And then there is an efflux program to remove the cholesterol then from the brain. And the system uses lipoproteins. And then the lipids resolve from the lesions, and inflammation goes away. Now, we have been looking at this process and compare this process in young and aged mice. And we do this because it was known that in aged mice repair is not as good as in young mice. And by looking at differences, we can find out key elements that are important for the remyelination. So I'm just summarizing some of the data in these cartoons. So the first phase after the lesion has been formed is the debris clearance phase. So you get the microglia in the lesion, they peak, and once they have performed their task and taken up myelin debris and metabolized myelin debris, then they resolve from the lesion and repair is initiated in the myelin repair phase. Now, when you look at this in aged mice, then the first phase, the debris clearance phase, looks quite normal. However, what was known before, that remyelination is impaired. And what we found is that the microglia remain in the lesion. There's persistent inflammation. There's a failure to resolve the inflammation in aged lesions. So what is the basis of this problem? Now, when we looked at the cells of the microglial cells in aged lesions by electron microscopy, we identified that these cells appear as foam cells. So they are full of lipid droplets, as shown here with these white structures. And there are cholesterol crystals that form. You see crystals here in lysosomes, and the majority of the crystals, in fact, form on the lipid droplets. So this shows that there could be a problem of handling lipids in aged lesions. Now, what we identified was that there was an impairment to induce the reverse cholesterol pathway in aged microglia. Now, the reverse cholesterol pathways depends on the liver receptor transcription factors. So these are nuclear transcription factors that sense oxysterols. When they have done this, they induce a transcriptional program that depend on ApoE and ABCA1 and ABCG1. So ABCA1 and G1 are transporters that are localized on the plasma membrane, and ApoE absorbs the lipids from these transporters from the surface. Now, what we found is that these LXR target genes were insufficiently activated in microglia in aged mice. Now, when we used LXR agonists to improve the response of the LXR transcription factors, we could fully rescue lesions of aged mice. So here you see the lesion volume in young mice at 3 months of age, and in aged mice, you see the poor recovery of the lesions. However, when we add in these aged mice the LXR agonist, we can fully rescue the repair process. So what we have seen is that there seems to be a problem to induce the reverse cholesterol pathway, and we have seen that this problem induces inflammation in the lesion and subsequently oligodendrocyte precursor cells and do not differentiate into remyelinating oligodendrocyte. Now, this process that we identified reminds us of arteriosclerotic lesions, where you have a similar problem. So you have macrophages in the blood vessels, and you have LDL cholesterol, which is oxidized, which is taken up with time in these macrophages. And when the lesions progress, there's inflammation in this lesion and plaque rupture and so on. So based on these analogies, we thought maybe there are the risk factors that lead to arteriosclerosis might be shared and also would lead to poor lesion recovery after demyelinating injury. So motivated by this, we used mice and fed these mice with a Western diet. That is a diet that we all consume, that is high fat and high sugar diet. It's high in fat, as I said, so, contains also a high proportion of saturated lipids, and we have a high proportion of sugar. Now, when you use this Western diet and control diet, and then you do a lipidomics analysis of the plasma, then you can clearly see, as expected, how the different control diet and Western diet separate. And as indicated or from the content of the diet, we see that the lipid plasma lipidome contains a high amount of cholesterol esters, a lot of saturated lipids, and chain lengths are in general shorter. Now, this was expected. What was not expected was that the lipidome also changes in the brain. So we analyzed the lipidome of the white and the gray matter, and they separate here as expected. in the control diet, but we see that the mice that had been fed with a Western diet have a different lipidome compared to control diet. And again, we found more saturated lipids and the shorter chain lengths in general. Now, this was surprising because there is a blood-brain barrier that is impermeable to lipoprotein transport. Now, when we look at the mice that we have put on a Western diet, then within a few weeks, they gain weight. So after 4 to 6 weeks, you can see a significant change in weight. But there are also many other things that happen in these mice. So the glucose tolerance test becomes pathological. So they develop diabetes after a few weeks. So they have a whole lot of metabolic disturbances. And we were interested to see how do these animals repair lesions. So we induced stereotactic lesions in the white matter, as I described previously, and then we looked at first at the demyelination and then at the recovery phase. Now, for time reason, I will just summarize the data in a cartoon. So in a lean mouse, myelin is taken up, metabolized, then the lipids resolve and induce, when the microglia leave the lesions, then they induce a repair program. However, in the obese mice, we found similar as in the aged mice that lipids are accumulated in the microglia. We see more foam cells, and we see an impairment of resolution of inflammation, and the whole downstream repair process was disturbed. So this was quite surprising that something very different, aging and obese, leads to a similar outcome. And we also found that treating these mice with LXR agonist could completely rescue the phenotype. Now, there were some differences between aged and obese mice, but in general, the pathology had very many similarities. So this raises the question, what is the underlying reason for the failure of microglia to clear lipids in lesions of aged or obese mice? Now, I told you that there must be a system that senses the damage and then translates this information to an effector program that initiates a repair program. So it's possible that in aged or obese mice, there would be inefficient sensing. This could lead to reduced responses and effector programs. There could also be a primary problem of the response program. Now, to begin to address this question, we looked at different sensors, and one that we identified was TREM2. TREM2 is a key lipid sensor in microglia, so it's a well-studied molecule because it's implicated in Alzheimer's disease. And it's a ligand for anionic lipids, and we and others have shown that it also interacts with myelin debris, and it can interact with lipoproteins like ApoE. Now, once the ligand has bound to the receptor, it induces a signaling pathway based on PI3 signaling and other not so well understood signaling cascades. But at the end, there's a well-known response program, and the response program are genes transcript involved in phagocytosis and lipid metabolism. Now, the negative feedback of this pathway is a cleavage here by metalloproteases, which turns down the signaling. Now, we looked at the signaling pathway. So these are cell culture experiments. So in the upper panel, we have TREM2 in the cell lysate, and in the middle panel, you have secreted TREM2 in the culture medium. So when you incubate microglia in culture with myelin debris, then you see that it's the TREM2 is clean. So myelin turns down-regulates its own signaling pathway. Now, why is this important? Because I told you that when TREM2 is in an uncleaved form, then the signaling pathway that leads to increased lipid metabolism is activated. However, when it's cleaved, the signaling is shut down. So based on this, we thought it would be a therapeutic approach could be to prevent the cleavage. So we collaborated here with Christian Haas from our institute and Denali Therapeutics. So they had developed an antibody which prevents the cleavage of TREM2. So we used this antibody, And first we tested in cell culture, we indeed can prevent the cleavage that was induced by myelin debris. So you have here the increasing amount of soluble TREM2 in the supernatant, and here the control antibody. When we use the agonistic TREM2 antibody, we can completely prevent this cleavage. So the antibody was doing what it's supposed to do, and it could also increase the signaling downstream of the receptor. So therefore, we use this antibody in vivo to see, can we improve remyelination in vivo. So first we used the paradigm with Western diet. So we treated mice for 4 weeks with Western diet. We induced the lesion, then we did 3 IP injection with the agonistic 49 antibody to increase TREM2 signaling. And then we indeed could see that we can reduce the amount of foam cells in the lesions. So normally we have a higher amount of foam cells in lesions, but by activating the TREM pathway, we could rescue this phenotype. Now, similar findings were found when we used aged mice. So here we induced the lesion, again, try 3 IP injections using this agonistic antibody, reduced number of foam cells, and we also quantified the number of regenerated, remyelinated axon, and so an improvement when we use the TREM2 agonistic antibody. So what I showed you here is that we have identified a TREM2 as a sensing system, and this is consistent with also with other people's work, and this induces lipid clearance in the cells and leads to immune resolution and a better outcome. So there are now several antibodies that have been developed by different companies, and they are now entering into clinical translation, mainly now for Alzheimer's disease, but there are also trials planned for multiple sclerosis. So I showed you data that was completely based on mouse work. So the question is, what is the relevance of this for human multiple sclerosis? And is there any evidence that could be impaired lipid clearance in MS lesions? So Daniel Reich used MRI of autopsies to identify chronic active lesions. So I mentioned this were the worst type of lesion because you still have persistent inflammation in this lesion at the edge, and they correlate with progression. Now we did single nuclear sequencing of individual cells in this lesion edge and identified that these cells are microglia, and he called them microglia inflamed in MS because they were inflammatory activated. And in addition, lipid genes were upregulated, therefore they called them microglia foamy. So we think that this is, at the moment, the best evidence there could be also in humans a problem of handling lipids in chronic active lesions. Now, so far, I've shown you that there are foamy microglia, and I've shown you a correlation. Higher number of foamy microglia leads to poor regeneration. But I don't want to leave you with this concept, because it's possible that foamy microglia, in fact, is an adaptive response. So to study this, we used a knockout mouse where we knocked out an enzyme, the ACAT sort1, which is important to form cholesterol esters. And this is important to store cholesterol, free cholesterol, then in lipid droplets as esters. Now, when we use a knockout mouse, this ACAT knockout mouse, and we use lesions now in the spinal cord, then we see, as expected, these mice do not form almost any foam cells. However, these mice also do not show good recovery. So we have less remyelination and we have chronic inflammation in the lesions. So here we have a different setting. We have less foam cells, but a poor outcome. So how we have to see this is that foamy microglia, in fact, is an adaptive state. So it's important to store lipids safely into lipid droplets. But then it's also important to resolve these cells by the reverse cholesterol pathway and by the actions of lipoproteins. Now, if this does not happen, then these foamy microglia might convert into foamy inflamed microglia. And this is the maladaptive state that correlates with poor recovery. Now, it would be interesting to have the transcriptional profile to understand what is the difference between these two cell states. To begin to understand this, we have, together with Urs Götze, who has led this project, devised a spatial transcriptomics correlated electron microscopy platform. So we used MERFISH. This is a spatial transcriptomics technology that provides insight in the transcriptomes of the cells in the brain. However, it has no structural information. Therefore, we combined it with electron microscopy to bring together a transcriptional profile with structural information in lesions. Now, with MERFISH, you can, after performing this transcriptional profiling and cell segmentation, get the all the cell types of the brain and also some of its activated and different states. Now we use this technology and we induced a lesion. You can see the lesion here, but it's red color because these are transcripts that are high in microglia. So you can see here the lesion. And now we can correlate the spatial transcriptomics with electron microscopy. So we used an adjacent section very close to the spatial transcriptomics section and aligned these two sections. And then we could zoom in on the EM images and looked at structural components here and do morphological annotations of different cells. So I will not go through all the annotations here, but for the purpose of this talk, just focus on the foamy microglia. So these are easily identified in EM images because you have lots of lipid droplets in this microglia. And now we can identify in the lesion all the regions were identified by EM foamy microglia. And now we can ask what is close to these foamy microglia, what are the transcriptional responses of cells close to these microglia. And what we identified is that there are microglia that we call lipid-associated microglia, because if we look at the transcriptional profiles of these microglia, we see genes that are typical for form cells. So we see here genes that are important for forming lipid droplets or components of lipid droplets. And then we see the pathway that's important for reverse cholesterol export, like ABCA1 and G1 and ApoE. So now with this technology, we can bring together structural information with transcriptional information, and we can now begin to understand what is the difference between a lesion in a young and old animal. And more importantly, we can also use this technology, and we are starting to apply it in human autopsy sections. So with this, I want to close, just give you a very brief summary of the key message of this talk. If you have an acute demyelinating lesion, this induces what I call reparative inflammation that is important for the repair process. So we have shown that the lipid metabolism is important for the resolution of the microglia cells within the lesions. So if lipids remain in the cells, you have chronic inflammation and poor regeneration. And with this, I want to close and want to acknowledge the people in the lab that have done the work. The main person that started all of this was Hugo Cantuti, together with Dirk Hitzner, and then Mar Bosch followed up the project with Gary Guna. And I already told you about our collaboration with Urs Götze and his postdoc Peter Androvich, who were doing this spatial transcriptomics EM correlation, Christian Haas and Denali Therapeutics for providing us the agonistic TREM2 antibody, and Martina Schifferer for a lot of the EM work, which I have shown you. And with this, I'm at the end, and thank you very much for your attention.